Today we are going to learn about the concept of protection of staff working in diagnostic laboratories or medical microbiology laboratories. So there are various things which we can do to protect the staff from getting infections from various dangerous microorganisms which they are handling in the lab. So there are five major strategies for their protection. That includes immunization of the staff, training of the staff, health checkups, PPEs, that stands for personal protective equipments, and disinfectants. Right, so one by one we'll discuss about them. First we are starting with immunization. As you know, immunization is a very important prophylaxis measure to protect ourselves from any kind of infection. However, we do not have vaccines available against all the infections, but still we have large number of vaccines which is available for use. However, the people who are working in the lab, they must be given few vaccines before they are allowed to any laboratory work for diagnosis. So CDC has recommended vaccines for healthcare workers that includes hepatitis B vaccine, influenza vaccine, mumps, measles, rubella, which is MMR vaccine, varicella vaccine, tetanus, diphtheria, and pertussis, that comes in the form of DPT, and some places it is also called as TDAP. T stands for tetanus, D stands for diphtheria, AP stands for acellular pertussis vaccine. However, in India, we are mainly using TDP, that is P stands for whole cell pertussis vaccine, and meningococcal vaccines. So these are some of the recommended vaccines by WHO, CDC. You can go to the given link, this link, and you can download the entire document and study for more information. So immunization of staff with these vaccines will protect them from getting infection or from any of the microorganism which is responsible for causing these diseases. Other than this, if a laboratory staff is working on a specific microorganism, if vaccine is available, they could be given that particular vaccine also. For example, if somebody is working on Mycobacterium tuberculosis, those people might be given BCG vaccine before they allowed to work into that lab, and so on. Second important thing is training. First thing, I think we all are convinced that trained staff can save a lot of time. They can, because of this, they can save money. Their productivity is very good, their efficacy is very good. We can compete with trained staff much better, in a much better way. They always produce better results, they save time. So there are a lot of advantages of having trained staff. However, here we are talking in context of safety of the staff, right? So to keep the staff safe, we need to give them a lot of training. We need to make them experts of their work, and for that we have to give them training. Now obviously, when some staff is hired into an organization, they are already well educated and well trained. However, when they are joining a organization, there are several things which has to be taught to them, or they have to be trained for several new things. So we need to give these new staffs different trainings at the time of joining. So this kind of trainings are usually called as job specific trainings, which usually happens in the beginning of their joining. Nowadays, not only in medical laboratories, even if you go to software companies, they are providing initially 6 months or 1 year job specific trainings to their new employees. However, once your staff is experienced, they are working since some time, then they might not require job specific training. However, you will have to give them certain refresher trainings and certain rotational training so that they can learn all kind of work which is going on in the laboratory. Then, as our lab is progressing, you might understand some employee working in a laboratory since long time may not be well adapted to the new features or new instruments or new functions which is happening in the laboratory. So we might have to send these aging employees or old employees to advanced trainings so that they can cope up with new generations. More than this, all staffs working in the laboratory must be trained in two basic aspects uh, s- microbiology, that is biosafety and biosecurity. So these two trainings are must for any kind of microbiology laboratory staff. Other than this, other than this, these staff must be regularly evaluated for their performance and knowledge by conducting various tests or competitions, so that they always be alert when they are working in the lab. Next thing is regular health checkups. As you can understand, by this we can actually identify that all the staffs who is working with us are healthy, and if there are certain health issues with them, they can be given some break so that they can recover and join back the laboratory. It is very important. It can be done on monthly basis, or at least in once every 3 months. So this is important and should be done. Next very important thing is personal protective equipments. So according to Occupational Safety and Health Administration, PPE can be defined as specialized clothing or equipment worn by an employee for protection against infectious material. There are various types of PPEs which we can use, that includes gloves, gown and aprons, mask, eye goggles, overhead, foot protection. I will try to show you all of them one by one through various diagrams. So gloves could be of several types. One of the most popular and widely used drug gloves in medical microbiology labs are sterilized gloves. These gloves are made up of latex and they comes individually packed, as you can see in this wrap. This is a wrap of one pair of glove, right? And they are mostly used during surgery and they are use and throw. Then we have non-sterilized gloves. They're also called as powdered gloves. These gloves are also made up of latex and they are also disposable and single-use ones, right? Mostly this type of gloves are used in microbiology labs. We can use them, we can even disinfect them by using disinfectants, and then we can, after finishing our job, we can discard them. They are cheaper also and can be used effectively. Next is heavy duty gloves. They are reusable and we can even decontaminate them by autoclaving and by other many other procedures. They are basically used for handling medical waste. They are also used for performing environmental cleaning, and there are other advanced type of gloves are there which is used for handling liquid nitrogen. Sometimes the people are wearing them to transfer the materials to keep their hand protected from a damage. So like that we have different type of gloves which is used in laboratories. Other than this, we have aprons and gowns. As you can see in this diagram, I have shown you three kind of body coverings. One is aprons or lab coats. There are different designs available for apron which is covering large portion of our body is good usually. Then we have gowns, as you can see. So aprons are reusable. You can autoclave them, you can wash them, and you can reuse them. Gown and the hazmat suit are use and throw ones. Usually gowns are made up of cotton and they are permeable. However, nowadays we are having impermeable gowns also. Permeable gown are basically used where we just want to prevent any kind of contamination from body to product, like in vaccine industries or pharmaceutical industry or a food industry. However, whenever you want to handle a dangerous microorganism, you always has to use a impermeable gown. Better than this, we have hazmat suits which cover the entire body from hat to boot, and it is completely impermeable. So like that we have different types of aprons and gowns which can be used as per our requirements or as per the category of organism you are handling. Right now there is a outbreak of COVID-19, coronavirus in our country also. So you might have seen various scientists are uh, you know are wearing this kind of suits. So basically these scientists are wearing a hazmat suit which is the impermeable type, because they don't want this virus to get absorbed into the cloth of gown and enters to their own clothes which they are wearing. So hazmat suit is more suitable to handle this kind of samples. Next is eye goggles. As you can understand, it will protect anything to go inside our eyes, so we can use it. It is usually made up of plastic and it is also disposable one. Then we can use overhead to protect anything to enter into our hairs, and we also can use it to prevent our hairs to fall down into our samples or products. Mask, this is again a very important piece of PPE. Mask could be of various types, however two important masks are standard mask, which is also called as plate mask, or ply mask, and then we have N95 or N99 mask also. So standard masks can reduce the risk of getting infections from droplets. However, it is not very effective against aerosols. Right? Right now for COVID-19, as it has been identified, it's a droplet infection. That's why even a plate mask or ply mask or standard mask is quite effective against this. However, N95 mask provides you much better protection, obviously both against droplets and aerosols, and it can prevent approximately 95% of the particles, which is even less than 1 microns in the size. The pore size of N95 and N95 mask is less than 1 micron. So they are very good, and usually when you are handling any microorganism which are usually airborne, you are supposed to use N95 mask or N99 mask. Next is closed footwear. So we can use shoes, as you can see in diagram. However, shoe covers is not very much recommended, though many people are using, but shoe cover is not that much recommended. However, if closed footwears are not available, then we can use shoe covers. Usually that too particularly when we are on field jobs, field duties, as you might have seen various technicians roaming into the public settings to collect the samples from the people. You might have seen they are wearing shoe covers. So for outdoor jobs or for field jobs, even shoe covers can also be used. However, for laboratory work, shoe covers are not recommended. Next thing is positive pressure suit. Now one thing you need to understand is positive pressure suits are only used in BSL for laboratories, nowhere else. As you can see, these suits are they are made up of very robust material, very thick material, uh, which is like plastic like, and this suit is completely airtight suit, in which, as you can see, this tube like structure which is hanging over here. So this tube is attached to the suit, and this will start filling the air into the suit like how we are filling a balloon, and it will continuously filling the suit, and there are certain pores in the suit which will allow equal amount of air to go out from the suit. So always air circulation is maintained inside the suit, and suit remains swollen also, or like a ballooned also. Therefore, even if there is any leakage into the suit, no microorganisms can get inside the suit, because no air can get inside. If there is leakage, air will only go outside. Because of that, microorganism cannot go inside. That's why positive pressure suit is very good in BSL 4 settings. However, in any other lab it is not used. Air which is getting into the this positive pressure suit is filtered by HEPA filter, right? This positive pressure suit is also called as moon suit, space suit also, right? Next important mechanism of staff protection is use of disinfectants. Now all of you understand disinfectants are the chemicals or mixture of chemicals which is used for protecting the staff, which is used to reduce the number of microorganisms in which they are not infectious anymore. So a chemical or mixture of chemicals used to kill microorganisms but not necessarily their spores. Disinfectants are usually applied on inert surfaces or objects. Right? We can also clean our hands with disinfectants, as you are doing. You are using hand sanitizers nowadays, or you might have used 70% ethanol in lab, right? So in this first table you are seeing they all are chlorine based disinfectants, and they are very useful and very potent even against viruses. Therefore they are widely used for doing surface cleaning into the laboratories. You might have seen nowadays for because of this COVID-19 outbreak, various people are spraying some liquid like chemical onto some vehicles and on roads and surfaces. That is basically these chlorine based solutions only. In that too, it is specifically mostly it is sodium hypochlorite solution, right? So this kind of chemicals can be used. So different chlorine based solutions are available. If you want to disinfect a clean surface, like for example your table is looking clean, right? So that is called clean surface. Dirty surface means when it is not looking clean, right? So it is just the visual difference, a clean surface can also have microorganisms, right? So here right now we are talking about killing the microorganisms from a visibly looking clean surface, right? So you can prepare uh, these concentrations of these different chemicals, while when you want to clean dirty surfaces, then the concentration of these chemicals will be high as per this table. So you can choose this chemical accordingly and prepare. Other than but this chlorine based substance are only suitable for surface cleaning, not for hands or other things, instruments and all, right? Other than this, we can use formaldehyde as 5% liquid disinfectants. This is also very dangerous and very effective disinfectant. However, it causes skin irritation and eye burning, therefore it is not very much recommended. Glutaraldehyde can also be used, which is also having similar kind of side effects. However, this is also a very potent disinfectant. Phenolic compound like triclosans is common in product for hand washing. Like you are using this in Dettol, Sablon, they all are having phenolic compound like triclosan, right? Next thing, you can use alcohol, and this is the most widely used family of disinfectant, where you can use ethanol, you can use 2-propanol, which is also called as isopropyl alcohol at 70% concentration, which is quite effective. You can also use iodine, generally um, unsuitable for use on medical oblique dental devices. Hydrogen peroxide at 3% concentration can also be used for surface cleaning, not for personal use. So these are different disinfectants which you can use for cleaning the surfaces, cleaning the instruments, and cleaning your hands and other surfaces. Now these things will always help the staff members not to get exposed to various microorganisms which is there unknowingly. So I hope with these information you might be clear how staff members will remain safe while working in the lab. Obviously there are many more things which can be done, however these are some of the major aspect of staff protection. Thank you.